Okay, so in this PowerPoint presentation, we are going to cover the process of meiosis, chapter 11. We just finished chapter 10, so you've heard about mitosis. That's one type of cell division. This is the second type of cell division. It's called meiosis. And so we said that mitosis in animals, which humans are included, right, in that kingdom, mitosis is going to lead to the production of all of our somatic cells, our body cells. Now this process of meiosis, we're going to see, leads to the production of gametes, or sex cells. So egg and sperm cells are not produced by mitosis, instead they're produced by meiosis in animals, including humans. Now when we talk about plants towards the end of the semester, we're going to see that plants actually meiosis in plants produces spores, and then spores give rise to gametes. Okay, so it's a little bit different depending on the kingdom that we're looking at. So let's jump into meiosis. So when we talk about meiosis, we have to talk about reproduction. All living organisms are capable of reproduction. If you remember back to chapter 1, we went over the characteristics of living organisms, and being able to reproduce was one of the key characteristics found in all organisms. So all organisms are capable of reproduction, making more of themselves. Okay? Reproduction comes in two forms, or there's two types. There's asexual reproduction, and then there is sexual reproduction. In asexual reproduction, that's when you have one parent cell that literally just divides into two. So it's pretty much just mitosis, or if you remember that term, binary fission. The parent cell gives rise to what we say clones, or identical organisms. Okay, so if we take, for example, a prokaryote, like a bacteria, like E. coli, let's say, you have a parent E. coli cell. Its genetic material gets copied. It goes through binary fission, or mitosis, and you end up with two daughter cells that are genetically identical to each other, or we could say clones of the parent. Okay, so again, many unicellular prokaryotic organisms tend to reproduce asexually. Very few multicellular organisms will use that mode. Instead, most multicellular organisms, including plants, fungi, animals, we're going to reproduce sexually. So sexual reproduction is very different. In sexual reproduction, you have two parents that are going to participate. It's going to be the result of two haploid cells. Remember that term haploid means half of the genetic information, okay, found in a somatic cell. So you have two haploid cells carrying half the number of chromosomes. They're going to come together in a union called fertilization, and that's going to produce the first cell of the next generation, or the offspring. And that first cell is going to be called a zygote, and it will have that restored diploid chromosome number. Okay, so if we're talking about humans, for example, you know that all of your somatic cells have 46 chromosomes, or 23 pairs of homologous chromosomes. Egg and sperm cells have just 23. They don't have pairs of homologous chromosomes, so they just have 23 chromosomes each. When an egg with 23 chromosomes is fertilized by a sperm cell with 23 chromosomes, that diploid zygote now has 46 chromosomes, or 23 pairs, right? Two copies of each. Okay, so in sexual reproduction, you have that diploid chromosome number being restored in the zygote. It's really important, though, that those cells that are participating in that union are haploid. Okay? How are haploid cells produced? How are egg and sperm cells produced in humans? They're produced by the process of meiosis. Okay, so this is cell division in which chromosome number is reduced by half. Or we can also call it reduction division. And that's what the focus of this chapter is going to be on. We're going to go over the steps of that process. Why is meiosis so important? Again, to ensure that chromosome number stays the same generation after generation. If meiosis didn't occur, then chromosome number could double, and that would be very bad. Why is sexual reproduction important? Why is it favored in organisms that are able and capable of reproducing sexually? Well, sexual reproduction leads to enhanced variation. So think about it. You have two parents that are participating in the production of that offspring. It's not cloning, right? Offspring will not be genetically identical to either parent. Instead, the offspring is going to have traits, right, alleles, right, contributing from both parents. And therefore, they're going to look different. They're going to have variation in pretty much all of their traits, okay? Or the potential for variation in all of their traits. Why is that important? So it's important to enhance evolutionary success of the species. The more variation that's present in the species, the higher the likelihood that that species will survive. Okay? A lack of genetic diversity in a species is very bad. And lastly, again, most eukaryotic organisms, animals, plants, fungi, are going to participate in sexual reproduction. Some of those organisms, for example, there are some species of fungi that can reproduce sexually or asexually. And there's even some lower level animals that can also reproduce asexually. Okay, but by and large, for eukaryotic organisms, the preferred mode of reproduction is sexual. Okay, so here's our list of learning outcomes for the chapter. We're going to be able to describe the behavior of chromosomes during meiosis. So how are they moving compared to mitosis? Differences between meiosis 1 and 2, those meiotic divisions. We'll be able to describe the cellular events that take place during meiosis, the differences between meiosis and mitosis, and lastly, mechanisms that produce genetic variation. Okay, so that's where we're going in this chapter. So let's start off with some definitions, and I know that we did these in chapter 10, but I just want to reinforce them, make sure you understand them. So genetic material in a eukaryotic cell is going to be found in the nucleus, okay? And when it's in the nucleus, it's not just spread out naked, right? That would be dangerous for the DNA. Instead, it's in the form of chromatin. And so chromatin is going to be DNA plus, and by now you know that the protein that's going to be involved in organizing DNA are those histone proteins. Remember, DNA gets wrapped around a core of histone proteins. You end up with the beads on the string, right? Nucleosome followed by a linker DNA. And that's pretty much it for chromatin. Okay, there can be a little bit more of an organization with other proteins involved, but that's pretty much it. If we're looking in a cell before it divides, so an interphase cell, that DNA plus protein is going to be pretty diffuse, kind of spread out in the nucleus, but again, still associated with those proteins. So in an interphase cell, we don't have tightly condensed chromosomes. Instead, we have DNA organized by proteins. Okay, now the opposite of that, or the next level, I should say, of organization up, is an actual chromosome. So remember, a chromosome is going to be completely condensed DNA. So not only do we have nucleosomes, but those are going to be organized into that 30 nanometer fiber, which then get further coiled into completely condensed chromosomes. Remember that, let me just get my pen here. Prior to DNA replication, you would just have a one-armed chromosome, but after DNA replication, you have a two-armed chromosome joined here at the centromere. Okay, this one-armed chromosome is called a monad chromosome. It's one arm. And this chromosome here is called a dyad chromosome. And a dyad chromosome is the result of DNA replication, where we go from 1 to 2 sister chromatids. So we go from having 1 chromatid to 2 sister chromatids. Okay? These sister chromatids, remember, are going to be 100% genetically identical. They're the same size, shape, they have the same genetics, the same loci, the same genes in the same position, as well as the same alleles or forms of those genes. Okay? Now, in a diploid cell, which remember all of your somatic cells are diploid, and the cells that are going to be dividing here in meiosis, the parent cells, they start off diploid as well. Okay? Diploid cells, remember, have 2 sets of chromosomes. So we use 2N to represent diploid. Okay, they have a maternal set and a paternal set. And those sets are going to be homologous. So remember, you inherited 23 chromosomes from your mom, 23 chromosomes from your dad, and so all of your cells now have all 46 of those chromosomes. Chromosome 1 from mom and chromosome 1 from dad are a pair. They're a homologous pair, which means that they are the same size, shape, they have the same location of their centromere, and they have the same genes, but maybe not alleles. Okay, so before we move on, let me just draw a homologous pair. So this would be, let's say it's chromosome 1 in the dyad form. So this is after DNA replication, so we would be in meiosis right now. And I'm going to draw its pair now. Here's chromosome 1 from dad. I'm going to use a different color to represent that. Okay, you're going to be doing some of this in lab too. So look at these chromosomes. They're the same size, they're the same shape, they have the same location of their centromere. And if I were to look closely, let's just say, for example, let's say I looked right here at this genetic locus and I found the eye color gene. And let's say red represents what originally came from your mom. So the allele there, the gene for eye color is there. The allele that your mom gave you, let's say she gave you the allele for blue eyes, which we would represent, let's use the letter B. The letter B. Blue eyes is recessive, so I'm going to use a lowercase letter. I'm going to find the very same allele here, right? Because these are sister chromatids. So this must be a little b. Now when I look at the paternal chromosome, I'm going to find the same gene. So the eye color gene is going to be at the same location. Let's say dad gives you the allele for brown eyes. So the allele found here at this position is going to be a big B. And since this other sister chromatid, this other arm, is an exact copy, this is also going to be a big B. So this is a homologous pair. Yes, these chromosomes are the same size, shape, they have the same location of their centromere, they carry the same genes, but they may have the same or different forms of those genes. So either the same or different, we use the term alleles. Okay, but they are a homologous pair. So in our cells, we have 23 total homologous pairs. Okay, now that's not true of every species. Every species is going to have a different chromosome number, which makes them unique. For humans, our total number of chromosomes, again, is 46, but we have 23 homologous pairs in our diploid cells. Now, our haploid cells, which are produced by meiosis, they're going to contain half the genetic information. So again, for humans, that would be 23 chromosomes. And those 23 chromosomes would not be paired. There is no homologous pairs in a haploid cell. Instead, haploid cells only have one set of chromosomes, and we use 1N to represent that. Okay, so again, we're going to be getting into this process. So the cell that's going to undergo meiosis is going to start off as a diploid cell. By the end of meiosis, we have haploid cells. So again, for humans, our parent cell would have 46 total chromosomes, or 23 pairs. Our resulting cells are going to only have 23 chromosomes, no pairs. Okay? Okay. So we've said some of this. Let's just reinforce it. So again, that term fertilization is used in sexual reproduction. And what that term fertilization means, that's going to be the union or the coming together, the joining of two haploid cells from two different parents, so from two individual organisms. That's sexual reproduction. Again, that's fertilization. Now those two haploid cells, again, are going to contain just one set of chromosomes. We call those cells in humans, we call those gametes. Ooh, sorry. We're going to call those gametes. And those are going to include eggs if you're female and sperm if you're male. Okay, and there are many other species as well. We're going to call them gametes. We're going to talk about egg and sperm. I like to focus on humans because I think that it helps students if we relate to ourselves. Now the result of fertilization, remember, is that zygote. That's the first cell of the next generation. And that zygote is going to have two sets of chromosomes, right? One set from mom, or the egg cell, and one set from dad, or the sperm cell. Okay? We call the set from mom, we call those the maternal chromosomes, from the maternal gamete. And the chromosomes that are coming from dad, we call those paternal chromosomes, coming from the paternal gamete, the sperm cell. And the zygote, remember again, is going to have that restored diploid number, 2N, two sets of chromosomes. Again, most animals and plants are going to be diploid. In fact, most animals, the only haploid cells that there are egg and sperm cells. The rest of the life cycle of an animal is going to be composed of diploid cells. Now, plants are a little bit different. Plants will usually alternate between a haploid and diploid state, depending on the plant. One might be longer lived than the other. But again, plants that we're pretty familiar with are going to be diploid. We will see plants in the lab that do spend larger parts of their life cycle in the haploid condition. But again, for now, most animals and most plants are going to be diploid, containing two sets of chromosomes in their body cells, or somatic cells. All cells of a multicellular organism except their gametes. That's going to be those somatic cells. Okay? And remember, again, I gave you that definition here on the bottom of the slide for homologous chromosomes. When we have diploid cells, we have pairs of chromosomes. Those are called homologous chromosomes. Okay? Those chromosomes are going to be the same size, length, shape, same location of that centromere, and again, they're going to have the same genetic material, the same genetic loci, but they may have different forms or alleles. Okay, so let's get into meiosis. Move my camera down here. So meiosis. Meiosis is going to be cell division, okay? Nuclear division followed by cellular division, just like we saw in mitosis, except for here, you're going to go from one parent to 4 haploid cells. Remember back to mitosis, you went from one parent to 2 cells that were identical. Here, you're going to go from one parent to 4 cells that are not identical. Okay, very, very different. The parent will be diploid. The resulting cells will be haploid. So you go from one diploid cell to 4 haploid cells. Okay? Notice how we're going from diploid to haploid, which means that we're reducing chromosome number by half. So another name for meiosis is reduction division. And that term reduction refers to chromosomes being reduced by half. So again, in humans, we would be going from a parent cell that had 46 chromosomes to 4 haploid daughter cells that each had 23 chromosomes. Half the amount of genetic information that was contained in the parent cell, half the number of chromosomes. This process of meiosis, reduction division, is essential so that we can maintain chromosome number from one generation to the next using sexual reproduction. If this process didn't occur, then we would double chromosome number with each new generation, and it just can't happen, right? For humans, our chromosome number is 46. Anything more or less leads to problems, and a complete doubling of chromosomes would lead to huge problems, right? There would not be offspring if that were to happen. Okay, so here's our overview. So before meiosis occurs, we have interphase, just like we had before mitosis occurred. And interphase looks the same. There's G1, S, G2. Okay? The same things happen. Normal growth. The cell gears up for a division event. There's going to be replication of the DNA during S phase, right? We go from monad chromosomes to dyad chromosomes that are going to contain 2 sister chromatids. Okay, so interphase looks pretty much the same. Interphase will be followed by meiosis. And now meiosis is going to be split into 2 distinct parts. Meiosis 1, followed by meiosis 2. Okay, and luckily, for meiosis 1 and meiosis 2, there's going to be the same subphases. So we're going to see prophase, metaphase, anaphase, and telophase 1 in meiosis 1. And in meiosis 2, we're going to see prophase, metaphase, anaphase, and telophase 2. Okay, so the names are just like we already saw in mitosis, which is great, easy to remember. So let's look at meiosis 1. And we're going to look at both of these stages in more detail, but here's a summary. So in meiosis 1, what's going to happen is homologous chromosomes, they're going to come together, and then they are going to be separated from each other. They're going to end up in separate cells. So in humans, remember you have 46 chromosomes or 23 pairs. So what you would have is those 23 pairs pair up. And then those 23 pairs get separated, pulled apart, and put into different cells. So you just went from a parent cell that had 46 chromosomes to 2 cells that have 23 chromosomes apiece in them. Now those chromosomes are in the dyad form, and those 2 cells are going to then go into meiosis 2. But think about meiosis 1 already. You've already gone from diploid to haploid, from 46 to 23. Reduction division already happened during meiosis 1. In meiosis 2 looks identical to mitosis. You have those 2 cells with 23 dyad chromosomes apiece, and now the dyad chromosomes are going to be split at their centromere, and monad chromosomes will migrate to the poles. Okay, so during meiosis 1, you go from 1 cell that is diploid to 2 cells that are haploid, but their chromosomes are in the dyad form. Let me grab a different color. Dyad form. In meiosis 2, each of those cells gets split. Okay, these cells are going to be haploid, 1N. And if we were to look at chromosomes, they would be haploid, or just 1 arm. Okay, so we're going to make sense of this even more. We're going to go on and we're going to look at the individual stages of meiosis 1 and meiosis 2. Okay, what I just drew for you is here in a cartoon. Okay, it's an overview. So again, let's start off with this very first cell. I love how your book does this. So this very first cell is going to be a diploid cell, and for simplicity's sake, we're only drawing 2 chromosomes. Okay, remember inside of us, our cells, there would be 46 of these chromosomes, but that's a lot to look at. Okay, and in lab, when you're working through this, I'm going to ask you to draw, really simple cells as well, so that you can really visualize it without getting too confused. Okay, so here in interphase, our parent cell is diploid. Its diploid number is 2, so it has 2 chromosomes. Notice how the chromosomes are monad. because this is early interphase. And notice the color. So blue and red, perfect. Let's say that blue is the paternal and red is the maternal chromosome. So originally, the parents, right, of that parent cell gave those chromosomes. They're in the same cell now and they are a pair. Okay? Now during interphase, we're going to have DNA replication. And DNA replication is going to take us from the monad form of those chromosomes to the dyad form. So each of those chromosomes now will have two arms or will have sister chromatids. Okay? So here we have a homologous pair of chromosomes, and if you look at each chromosome, it's in the dyad form. So again, our chromosomes just duplicated. Okay, that's interphase. Now, during meiosis 1, what's going to happen is the homologous chromosomes are going to be pulled into different poles, different sides of that cell, and eventually they're going to be in different cells. So this right here and this right here, those two cells, are the result of meiosis 1. So homologous pairs were pulled apart and they end up in separate cells. Now, let's take a look here. Look at the parent cell, way up here. We said that its diploid number is 2 chromosomes. How many chromosomes are in each of these cells at the end of meiosis 1? This cell has 1 chromosome. It's in the dyad form, but it's just 1 chromosome. And this cell right here has 1 chromosome. So we just went from 2 chromosomes to 1. We already reduced it. Reduction division has happened. We reduced chromosome number by half. So this parent up here is diploid, and these two cells are haploid. Okay? Their chromosomes are still in the dyad form, and so we need to separate those. We need to go back to the monad form. Okay? And so that's what's going to happen in meiosis 2. We're going to separate sister chromatids. And so for this cell, we're going to split into 2, and each of those daughter cells will have a single monad chromosome. They're still haploid. Okay? And the same thing is going to happen here in this cell. We're going to split it into 2. So again, each of those daughter cells are haploid with 1 monad chromosome apiece. So by the end of meiosis 2, we have 4 cells. Okay, and again, each of those cells is haploid, having 1 chromosome, and that chromosome is in the monad form, 1 arm. Okay. Before we go on, I'm going to ask you just a couple of questions. So first question, meiosis usually produces blank daughter cells, 2 haploid daughter cells, 2 diploid daughter cells, 4 haploid daughter cells, or in yellow, 4 diploid daughter cells. So what is the end result of meiosis? Hopefully you answered in green there. The end result, by the end of meiosis 2, we have 4 haploid daughter cells. Okay? At the end of meiosis, chromosomes are in this form. At the end of meiosis 2, are chromosomes in the monad form, the dyad form, the tetrad form, or the diploid form? So again, hopefully you answered monad. Okay? Chromosomes are in the monad form at the end of meiosis. Okay, just wanted to make sure we were on the same page before we went on. So now let's get into it. So now we're going to go through the steps of meiosis 1, and then we're going to go through the steps of meiosis 2. Okay? Before we get into meiosis 1, again, remember that preceding meiosis, if we're looking at the cell cycle, we've got interphase, just like we did for mitosis. So interphase is going to come before this process. We have DNA replication that's going to occur during S phase. By the end of interphase, chromosomes are in the dyad form, consisting of 2 sister chromatids held together by the centromere. Remember, there's those cohesin proteins at the centromere. So again, we go from monad chromosomes to dyad chromosomes during interphase. The dyad chromosome, remember, is held together right here by cohesin proteins. That's the centromere. And if we take a look at the dyad chromosome, the 2 arms, arm 1 and 2, are sister chromatids, which means that they are exact identical copies of each other. Those were produced during S phase of interphase. Okay, so by the start of meiosis, we've got dyad chromosomes. Okay, so let's talk about prophase 1. So we're in meiosis 1. Prophase 1, P stands for preparation. The cell's going to now get ready for the first division, where we go from 1 cell to 2. So what's going to happen during prophase 1? Very similar to what we saw in mitosis. So first off, the nuclear envelope starts to break down, right? The centrosome is going to move towards the poles, and that meiotic spindle, remember we called it the mitotic spindle in mitosis, now we're calling it the meiotic because now we're in meiosis. The meiotic spindle is now going to assemble at each poles. It looks the same, right? If it's an animal cell, it's going to have these centrioles that we're showing here at the center of the centrosome, and the mitotic spindle is going to be composed of microtubules. Okay? Okay. What else happens? Chromosomes condense. They're going to become visible. They're going to be in that dyad form. And then this is something new. So what's going to happen is those homologous pairs of chromosomes are going to find each other, and they are going to attach to each other, forming these structures called tetrads. Okay, we say that they synapse, which means that they find each other and they join together. In this picture down here, we're showing that. So we actually have 2 homologous pairs, and each of those pairs, of course, has 2 chromosomes. We're showing the colors red or pink to represent 1 chromosome and blue to represent the other chromosome of the pair. Okay, so let's see. Okay, I'll use my red. So what a homologous pair looks like is this. Here's 1 chromosome of a homologous pair, and here's the other. Okay, they're going to find each other, and they're going to synapse. They're going to touch and cross and maybe even, here it is. So as they're crossing over, non-sister chromatids are crossing over. Where they cross is called a chiasmata. And there can be an exchange of genetic material. So the non-sister chromatid arms can actually swap equal amounts of genetic information, which is pretty neat. That can lead to enhanced genetic variation in gametes and therefore in offspring. Okay, I'm showing you this. We're going to talk a little bit about it, but we're going to really get into that deep if you take General Biology 2 with me. So we'll start off that semester, General Bio 2, with really the same chapter. So we'll cover meiosis, but we'll cover it in a little bit more depth. And we'll talk a lot about crossing over. And when we get into genetics, we'll talk even more about crossing over. Okay? Okay, so here's a nice picture of a homologous pair of chromosomes. This is 1 pair, right? We've got 1 red dyad chromosome and 1 blue dyad chromosome. They are a pair, a homologous pair. They're the same size, shape, they have the same location of their centromere, and they have the same genetic loci, maybe different alleles, maybe the same. Okay, what they're going to do is they're going to find each other, and they are going to synapse, connect. And there's going to be all kinds of proteins involved here that are going to hold these chromosomes together. Now, when this happens, we call this the tetrad form. Okay, so you're going to hear that term, tetrad. and tetrad meaning 4, okay? And so here, if you take a close look, each of the chromosomes of the homologous pair had 2 chromatids, or arms. And so if they join up, we have a total of 4 chromatids, or 4 arms. So a tetrad is a 4-armed chromosome. It's the result of a homologous pair of chromosomes coming together. It's synapsin. So that's going to happen during prophase 1 of meiosis 1. So again, we're simplifying things. We're showing 1 or 2 chromosomes, but remember, inside of humans, you have 23 pairs of chromosomes, and so there would be 23 tetrads that are going to form during prophase 1 of meiosis 1. Okay? Okay, and here's just a little image. Again, we would get into this more in general bio too, but just to show you crossing over. So again, if you take a look at this first figure, we've got a homologous pair of chromosomes, same size, same shape, same location of centromere, same genes. Let's look close though. They have different forms of genes A, B, and C in this case. So the blue chromosome has the dominant form of the gene, okay, the dominant allele for genes A, B, and C. If you look at the red chromosome, the recessive forms of genes A, B, and C are present. Okay, they're going to find each other during prophase 1 of meiosis 1. They're going to synapse. They're going to form a tetrad, and maybe they're going to have some crossing over or recombination occur. If there is crossing over and there is an actual genetic exchange that occurs, then look at the result down here. We've swapped the dominant form of gene C with the recessive form of gene C on those non-sister chromatids. So do you see this right here? Okay, that's called a recombination event, the result of a crossing over between 2 non-sister chromatids during prophase 1 of meiosis 1 when they were in the tetrad form. Think about the implications of that. It leads to more genetic diversity. Okay, so not only are you a mix of the alleles for each gene from your parents, but your actual chromosomes could end up with a mixture of alleles, and so that could just lead to even more enhanced variation. Okay, so you got sexual reproduction leads to variation. You've got recombination, which could also lead to variation. Variation is really important in an evolutionary sense. Okay, so that's prophase. So prometaphase now looks very similar to prometaphase of mitosis. Again, this is called prometaphase 1. What's going to happen is those microtubules forming that mitotic spindle, they're going to grow, and they're going to attach on each side of each homologous pair to the kinetochore of those chromosomes. So the kinetochore, remember, those are just proteins that are present at the centromere. And so the mitotic spindles at each pole, now they grow, and they're going to attach on each side of each chromosome pair. So if we look here in this figure, here's those mitotic, Here, let me get my pen. Here are those mitotic spindles. Oh, it's not letting me draw. Okay, now here's those mitotic spindles, right, growing out from the centrosome. Here's one pair of chromosomes, one homologous pair. Here's another homologous pair of chromosomes. Look, crossover did happen. Okay, we can see that exchange of color. It's pretty neat. And what we see now are the spindle fibers attaching on each side of our chromosome pair. Okay, and then what happens next is those chromosome pairs just line up in the middle. So metaphase 1, we have homologous pairs of chromosomes in the form of tetrads, 4-armed chromosomes. They just now line up in the middle of the cell. So again, in the cell pictured here, there's only 2 pairs. In our cells, there would be 23 pairs lined up in single file pretty much down the middle of the cell. Again, we can call that the midline, the equatorial plate, those are all correct. Okay, now how they line up, the positioning of the maternal and the paternal chromosomes, the colors here, that's random. Now you could have all red or maternal chromosomes on one side of that plate and all blue on the other. Or, you can have some on each side. That's a random event. You're going to learn about that again in General Bio 2 if you decide to take it. The random alignment of tetrads in the middle of the cell during metaphase 1 is called independent assortment. And that's really, really important. This is one of Mendel's principles. So when you talk about genetics, Gregor Mendel, the father of heredity, he discovered this law of independent assortment. So again, what that means is that there's two possibilities. Tetrads could line up like this or like that. Okay, and my hand's here. This is my hand, representing the chromosome from mom. Here's my hand representing the chromosome from dad. Dad could be on this side or on that side. And that's random, how they orient themselves. Okay, so in humans, again, we have 46 chromosomes, 23 pairs. Those 23 pairs can each line up in two different ways. And so let's calculate the possible genetic variation now that we can find in gametes due to just how they line up. It would be 2 to the n, and that n would equal the number of homologous pairs. So for humans, that's 23. So 2 to the 23rd power, that's over 8 million possible gametes that could be formed. And that doesn't take into account the potential for crossing over in genetic exchange or recombination. So sexual reproduction leads to really huge enhanced genetic variation. This is why you don't look identical to either of your parents or to any of your siblings if you have siblings, okay? Unless you're identical twins. It's different. Okay, so this is where we get genetic variation. We get it through sexual reproduction, we get it through independent assortment of chromosomes, and we get it through recombination events. Okay, and remember again, this is a little bit beyond what I need you to know for General Bio 1. When we get into General Bio 2, this will all come back. Okay, I'm just setting a foundation for you. Okay, this is just a figure showing you the two different chromosomal arrangements that can result during metaphase 1 and what that means. So in metaphase 1, if chromosomes lined up like this, well, look at the colors. So red's on the right-hand side and blue's on the left-hand side. So your red chromosomes, right, there's two different types here. We'll call it chromosome pair number 1 and chromosome pair number 2. Chromosome pair 1 and 2, the red chromosomes from each of those pairs would end up in one cell, and blue chromosome 1 and 2 would end up in the other cell. And so if you look at gametes, look. Okay, that's that genetic arrangement. Well, what if they lined up differently? What if we look at pair number 2, what if we flip-flop those? Well, then you get a totally different set of chromosomes in those end cells. The process is still the same, right? But you end up with, because of that different arrangement, you end up with different genetic variation in those gametes in the end. Okay, so it's really important to take into account how those chromosomes line up. Okay, so prophase 1, so first off, interphase, you had DNA replication. You go from monads to dyads. Prophase 1, you've got the chromosomes condense and they form tetrads. You might get recombination. Nucleus goes away. Centrioles move towards the poles. Okay. Metaphase, homologous pairs of chromosomes or tetrads line up on the metaphase plate. And now in anaphase, what's going to happen is those dyad chromosomes as part of those tetrads are going to be pulled apart. So we're separating the homologous pairs. And that's what's shown here in this figure. Okay, we're pulling those chromosomes. These are being pulled that way. And these chromosomes are being pulled that way. So we're separating the homologous pairs. If we go to telophase and cytokinesis, right, what's going to happen is those chromosomes are completely pushed to the poles. they may start to decondense a little bit. Nuclear envelopes reform. We have two. Okay, two nuclei. And cytokinesis is just going to divvy up the rest of the cytoplasm and organelles, and we're going to end up completely separating those two cells. So the end result here is two cells, 1, 2. Okay, and if we take a peek at those cells, chromosome number in each of them is now just 2. If you think back to the parent, and here let's flip back to the parent. Oh, here. If we were to count up those chromosomes here, there would be 4. 4 dyad chromosomes. And now in this cell, these 2 cells, each of them only have 2 dyad chromosomes. So we went from a diploid parent cell to 2 haploid cells. We just cut chromosome number by half. Okay, so that's the end of meiosis 1. So before we move on, go ahead and hit pause. Let's make sure you got this. You're going to go ahead and you're going to define those terms. You're going to talk about why meiosis is called reduction division. You're going to tell me what happens during interphase. You're going to describe prophase 1, metaphase 1, anaphase 1, and telophase 1. And then you're going to tell me what is the result of cytokinesis. So go ahead and take a break. When you're finished, come on back. I have some questions for you. Okay, so the first question here for you. Meiosis is also known as reduction division because, is it because the nucleus is reduced by half? Is it because chromosome number is reduced by half? Or is it because the parent cell shrinks during the process? Or is it because the amount of genetic material doubles? So why is meiosis called reduction division? So hopefully you answered because chromosome number is reduced by half. Okay. Second question. Chromosomes are duplicated during which phase of the cell cycle? Do chromosomes get duplicated or copied or replicated during prophase 1, metaphase 1, S phase of interphase, or interkinesis? So chromosomes get copied during S phase of interphase. Remember that S stands for synthesis, and that's DNA synthesis. Another question for you. All of the following events take place during prophase 1 except, so in pink here, homologous chromosomes pair up forming tetrads. Blue, the nuclear envelope breaks down. Green, the meiotic spindle is assembled. Or yellow, tetrads line up on the metaphase plate. So all of these take place during prophase 1 except, hopefully you answered yellow. So remember, tetrads lining up on the metaphase plate, that happens during metaphase, not during prophase. So during metaphase 1, right, because that term tetrads is there. Okay, so we finished meiosis 1. We're going to go on to meiosis 2. And so between meiosis 1 and 2, sometimes in some species, you're going to have a little bit of a break or a gap. Now again, this sometimes is long and sometimes it isn't there at all. It depends on the species that we're looking at. If it's there, that gap between meiosis 1 and the beginning of meiosis 2 is called interkinesis. There's really nothing that happens during interkinesis, nothing substantial. Sometimes students will see inter and automatically think DNA replication, and that's not the case. So during interkinesis, I want you to remember, pretty much nothing happens, and especially DNA replication does not occur. Okay, it's a really common mistake. So let's jump right into the stages. So following interkinesis, if it's present, both cells will enter prophase 1. Prophase 2. In prophase 2, pretty much the same thing happens that we see in regular old prophase when we're talking about mitosis. Okay, so chromosomes condense, if they decondensed at all, into the dyad form. The nuclear envelope breaks down and the spindle forms. That's pretty much it. Then we move into prometaphase/metaphase 2. And what's going to happen here are the spindle fibers will attach to the kinetochore of each dyad chromosome. And the dyad chromosomes will line up in the center of the cell. And so what I have here in our figure for us are 2 cells here. These are our 2 cells pretty much in metaphase. Okay? And so you'll see in both cells, they have 1 chromosome of each original homologous pair. And in total, they have 2 chromosomes. So each cell has 2 chromosomes, 1 chromosome from each original homologous pair. We notice the length. So these chromosomes here are shorter, right? So they were a homologous pair. And these chromosomes that are longer, they were a homologous pair. So those were separated. Okay? So now here, again, they're lining up in single file as dyad chromosomes. What do we think is going to happen? They're going to be split down the middle and down the middle. Okay, let's watch that. So metaphase 2, they line up. So here we are. In anaphase 2, what's going to happen is those centromeres, those cohesion proteins are going to break down. Centromeres are going to allow the sister chromatids to separate. Okay, so sister chromatids are pulled in opposite directions. So, for example, this chromosome right here just looked like that. Okay, this arm's going that way and that arm's going that way. They're being pulled apart. Okay, that's going to happen in both cells. So again, anaphase away. So anaphase 2, sister chromatids are pulled in opposite directions. And then telophase 2/cytokinesis 2. Those monad chromosomes are pulled all the way to the poles. They may start to decondense a little bit. Nuclear envelopes will form. So again, this was 1 cell, and this was our 2nd cell. And now they're splitting, and so we're going to end up, in the end, with 4 cells. Remember, the cytoplasmic separation happens during cytokinesis. Okay? That's it. So you can see that meiosis 2 really is the same as mitosis. But meiosis 1 looks pretty different. Okay? So let's compare meiosis 1 and meiosis 2. If we're looking at, let's say, metaphase, or here it says prometaphase, and anaphase, let's look at meiosis 1 first. In meiosis 1, we have homologous pairs of chromosomes, or tetrads, that line up. And then we have dyad chromosomes that are pulled apart. Where in meiosis 2, we don't have tetrads, we don't have homologous pairs. Instead, we just have dyad chromosomes that are pulled apart, and the sister chromatids, or monads, will end up in different cells. Okay? So we go from 1 cell, let me just erase this. We go here from 1 cell that is diploid to the end. We have 2 cells that are haploid. We start off meiosis 2 with our haploid cells, and we end meiosis 2 with haploid cells. Okay, let's put some numbers on this. So there was 1 diploid cell to start off meiosis 1. We have 2 haploid cells at the end of meiosis 1. We start off meiosis 2 with 2 haploid cells, and we end meiosis 2 with 4 haploid cells. So in total, we go from 1 diploid cell to 4 haploid cells. Reduction division. Okay, here's a summary. I'm not going to read this to you, but it goes over each step, gives you a picture of what a cell would look like, and it gives you the outcome. Okay, so go ahead and take a peek at that. And then here is another figure from your textbook where we are just comparing and contrasting mitosis and meiosis. So at the top here, we have meiosis. Oh. At the top, we have meiosis, and at the bottom, we have mitosis. And here's some comparisons that happen. So first off, in interphase, whether we're talking about mitosis or meiosis, we get DNA replication, and it occurs in S phase of interphase. So that's a similarity. Okay? If we look at meiosis now, meiosis 1 happens in meiosis, right? But we don't have a mitosis 1 and 2. So there is no, split or 2 phases. Okay? Synapsis of homologous chromosomes happens here in prophase 1 of meiosis 1. It doesn't occur anywhere in mitosis. Okay? So synapsis happens in meiosis, but it doesn't occur during mitosis. Crossover or recombination, if it's going to happen, it happens during prophase 1 of meiosis. It does not occur during mitosis. So there is no genetic exchange during mitosis. Homologous chromosomes lining up on the metaphase plate. Well, we only have that occurring in meiosis, and that's going to occur during metaphase 1. It does not occur during mitosis. In mitosis, we have homologous chromosomes, but they never pair up. They never get separated. Okay? Sister chromatids lining up on the metaphase plate as dyads. That's going to happen during metaphase 2 of meiosis. It happens just during metaphase of mitosis. Okay? And if you look really closely at meiosis 2 and you compare it to mitosis, they look the same. So meiosis 2 and mitosis are pretty much the same process. Okay, how about the outcome? So the number and genetic composition of the daughter cells produced. So in meiosis, we're going to go from 1 diploid parent cell. I want to highlight this. So in meiosis, you're going to go from 1. Hold on. Okay. You're going to go from 1 diploid parent to 4 haploid daughter cells. Look at the genetic material. We started off with 4 chromosomes in the parent. Now each one of these cells only has 2 chromosomes. So again, we go from diploid to haploid. 1 diploid cell to 4 haploid cells. Look at mitosis. You start off with 1 parent cell that is diploid, also contains 4 chromosomes. And if you look at the result, the daughter cells, there's 2, but each of them still have 4 chromosomes. So chromosome number is retained in mitosis, where chromosome number is cut in half in meiosis. Okay. Some other things to talk about. If we look at the machinery, the cell's machinery involved in this process, there's still a spindle. In animal cells, there would still be centrioles, and those spindle fibers are still made up of microtubules, whether you're talking about mitosis or meiosis. Those microtubules will still join at the kinetochore, those proteins present at the centromere region of chromosomes. Those types of things are still the same. How cells split is the same. So again, mitosis or meiosis, when you get to cytokinesis, if we're talking about an animal cell, you're going to have the cytoskeleton involved in a pinching mechanism that creates a cleavage furrow. If we're talking about plant cells, there's going to be that cell plate. So all of those things are the same. It's just how chromosomes interact with each other, and how many chromosomes end up in each cell in the end, and how many cells are produced. That's going to be the key differences here. Remember again, mitosis is used to produce somatic cells, all body cells in humans, and meiosis is only going to be used to produce gametes. Remember that goes back to sexual reproduction. In order to maintain chromosome number generation after generation, sex cells, egg and sperm cells, have to contain only half of the genetic information found in all the other cells in the body. And that's so that when the zygote is formed, it has the proper number of chromosomes. So again, for humans, that would be 46, or 23 pairs. And so therefore, our gametes can only have half of that number, just 23 chromosomes and no pairs. Okay? Okay, so that's all the talking I'm going to do. That's enough. So go ahead and answer these questions. So again, you're going to describe the events of meiosis 2. How many cells are produced? Are they haploid or diploid? Sketching here would be fantastic. If you can draw a cell, you got it. You're going to be drawing in lab too. So, and then you're going to compare and contrast mitosis and meiosis. So you're going to make sure that you show both the similarities as well as the differences between those processes. When you're finished, I just have a couple questions left for you. Okay. Okay, so the first one, the first one. If a muscle cell of a typical organism has 23 chromosomes, so a muscle cell, that's going to be a somatic cell, how many chromosomes will be in a gamete of that organism? So you've got 32 chromosomes in body cells. How many chromosomes will be in that organism's gametes? 8, 16, 32, or 64. And so hopefully you answered 16. Okay, so we've got to do reduction division. Meiosis is going to come into play here, and we're going to reduce chromosome number by half. Half of 32 is 16. Okay, next question. Which statement best describes the genetic content of the 2 daughter cells in prophase 2 of meiosis? Okay, so at the end of meiosis 1, you end up with 2 daughter cells. How would you describe those cells? Those 2 cells are now going to enter prophase 2. What are they going to be like? Is each cell going to be haploid with just 1 copy of each gene? Is each cell going to be haploid but with 2 copies of each gene? I should say of each dyad chromosome. That would sound better. Yeah, let's do that. So red is going to be haploid with 1 copy of each dyad chromosome, haploid with 2 copies of each dyad chromosome, diploid with 2 copies of each dyad chromosome, or diploid with 4 copies of each dyad chromosome. This one's a little tricky, the wording here. So hopefully you answered pink, right? Those 2 cells are going to both be haploid, and there's going to be no pairs of chromosomes. That's what I'm getting at. There's no homologous pairs of chromosomes. So they're going to be haploid with only 1 copy of each chromosome. Okay. Okay, next question. The phase of meiosis in which tetrads are separated and dyad chromosomes are pulled towards the poles is, does that occur in prophase 2, anaphase 1, anaphase 2, or metaphase 1? So which stage of meiosis are tetrads, first of all, going to be present, and second of all, pulled apart? Okay, when do tetrads get pulled apart into dyad chromosomes? So hopefully you answered anaphase 1. So first off, tetrads are only present in meiosis 1. They form in prophase 1, they line up in metaphase 1, and they get pulled apart during anaphase 1. Okay, tetrads are not present in meiosis 2. Okay, next question. What is a likely evolutionary advantage of sexual reproduction over asexual reproduction? So pink says, Sexual reproduction involves fewer steps. Or blue, there is a lower chance of using up the resources in a given environment. Green, sexual reproduction results in variation in the offspring. Or yellow, sexual reproduction is more cost effective. Okay, so hopefully you answered green. It's all about variation, right? So sexual reproduction leads to enhanced genetic variation, genetic differences in offspring. The more differences you have, the better the ability of that species to survive. And that's an evolutionary advantage. Okay. This is the last question. Which statement correctly describes the difference between monad and dyad chromosomes? Pink, monad chromosomes have 2 arms, where dyad chromosomes are single-armed. Blue, monad chromosomes are present in cells at the end of meiosis 1, where dyad chromosomes are present in cells at the end of meiosis 2. Okay, think about those two. Are either correct? Green, dyad chromosomes are made of 2 sister chromatids, where monad chromosomes are made of 1 chromatid. Or yellow, monad chromosomes are present in somatic cells. Dyad chromosomes are present in gametes. Lots of words here, so take your time in reading them. If you have to pause me, go ahead. Okay. So the answer here is green. Dyad chromosomes are made of 2 sister chromatids, identical, right, chromatids, where monad chromosomes are made of just 1 chromatid, 1 arm. Okay, that's it for chapter 11. So again, make sure you've got your Are You With Me's done. You want to go ahead and you want to upload those to Blackboard. Make sure that they're either in a Word or a PDF document, something that I can open on my end. And then you want to go ahead and you want to study, and you're going to go ahead and take the quiz. The quiz is a joint quiz consisting of questions from both chapters 10 and 11.